This episode of Cell and Gene, the podcast, is brought to you in partnership with Thermo Fisher Scientific and Applied Biosystems' new QualTrack real-time PCR and digital PCR solutions for biopharma. Give your cell and gene therapy development an edge with reliable and accurate qPCR and dPCR workflows backed by a trusted supplier. Explore the complete ecosystem of CGMP-compliant qPCR and dPCR assays, master mixes, and instruments at thermofisher.com/qPCR/biopharma. Hello, listeners, and welcome to this episode of Cell and Gene, the podcast. I'm your host, Erin Harris, and my guest for this episode is Dr. Michelle Mellion. She is the SVP, Head of Clinical Development at PepGen, a Cambridge, Massachusetts-based biotech focused on oligonucleotide therapeutics with the goal of transforming the treatment of severe neuromuscular and neurologic diseases. Michelle, thank you so much for your time. Thanks for being here.
Thank you, Erin, for having me. I'm really excited to be here to talk about our programs here at PepGen and how we're moving forward quickly.
Good. Well, that's actually my first question for you is I'd like for you to explain to Cell and Gene, the podcast listeners, one, PepGen's sort of company mission, and then talk us through your pipeline and what's really going on.
Yeah, so PepGen is a clinical stage biotechnology company. And basically what we are doing is we are creating a pipeline of disease-modifying therapeutics with the potential to safely and effectively target the root cause of serious genetic diseases. And so we're starting in, as you said, in neuromuscular diseases. And basically the premise of our technology is based on oligonucleotide therapies. We know that oligonucleotides can be effective for treating many different diseases. However, the challenges with oligonucleotides is that it's very difficult to get them to the tissues of interest. And specifically in neuromuscular diseases, obviously we're interested in muscle, smooth muscle, the heart. And for many of these neuromuscular diseases, we are also interested in getting oligonucleotides or therapeutic oligonucleotides to the brain. But because of the pharmacokinetic profiles of these oligonucleotides, they're quickly degraded in the plasma, and it can be very difficult to get them into those tissues of interest. So PepGen has focused on at delivering an effective concentration or clinically relevant concentrations to the tissues of interest with the use of their cell-penetrating peptides, and these are proprietary cell-penetrating peptides that are essentially conjugated to the therapeutic oligonucleotides. And what we have seen in our preclinical studies, as well as now in our recent completion of our phase 1 clinical studies, is what we have seen is that we do, in fact, have broad distribution to these tissues of interest, including skeletal muscle, smooth muscle, the heart, and we do see some distribution to the brain. And this has really supported our current pipeline, in which we are focused on delivering transformative therapy for boys and young men living with Duchenne muscular dystrophy, as well as we have a close follow-on program in myotonic dystrophy type 1. And not only will we be focusing on Duchenne muscular dystrophy, specifically looking at exon 51 skipping. So boys with Duchenne muscular dystrophy can have different mutations that are amenable to different types of oligonucleotide therapies. We are also evaluating and having discovery programs for exons 53, 44, and 45.
Excellent. Okay. And that's actually where I wanted to go next. And I was hoping we could talk through in a little bit more detail the trial for PGN-EDO51, which you said for the treatment of DMD patients whose mutations are amenable to exon 51 skipping approach. Can you talk us through that in detail? Where are we? What's next? Anything you can offer?
Yeah, absolutely. So we are very excited about the results of this phase 1 clinical trial in healthy volunteers. And basically, this trial was designed to inform PepGen about the potential safety profile of PGN-EDO51, because this was our first in human study with PGN-EDO51, as well as tell us what doses might be clinically relevant, so that when we went into patients, which we plan to go into DMD patients next year, in the first half of next year, we could start out at those therapeutically or clinically relevant doses and kind of skip over those doses that may not necessarily confer any clinical benefit. And so this study is a single ascending dose study in healthy male volunteers. It is consisted of cohorts of 8 healthy volunteers per cohort that were randomized 3-to-1 to EDO51 or to placebo. And these healthy volunteers received a single dose of EDO51 and then underwent muscle needle biopsies of their biceps at day 10 and day 28. And what we learned from the analysis of the data in this study is that we were able to achieve the highest level of exon skipping and highest level of oligonucleotide delivery to muscle after a single dose based on what is available in the public domain. And so this high level of exon skipping, what we saw was from the lowest dose at 5 mg per kg was up to 0.38%, up to our 15 mg per kg dose, where we achieved 2% exon skipping. And what has been seen in other studies is that what has been achieved in these other studies is 0.18%. So we are several-fold higher than what was seen previously. And this is very exciting, because the more exon skipping that you see, the more dystrophin production you are likely to get. The other thing that was interesting about our exon skipping data was not only did we see high levels of exon skipping at day 10, but we saw persistent levels of exon skipping at day 28. So when we went from day 10 to day 28, we actually saw higher levels, and we went from 1.1% to 1.4% at that 10 mg per kg dose. And what this means is that it tells us about the potential for accumulation with repeat dosing. So if we are able to see that there is an accumulation at the end of 1 month to 1.4%, we can imagine that over several doses that amount of exon skipping will continue to increase and deliver clinically meaningful levels of exon, or potentially clinically meaningful levels of exon skipping, which will result in increased dystrophin production. And so based on what we know from publicly available data, what we have seen from other studies is that there has been a relative increase over that period of time, and we anticipate that same increase. In terms of the dosing, we saw the highest level of oligonucleotide delivery to muscle that has been seen based on publicly available data. And the fact that we are able to get these clinically relevant concentrations into the tissues of interest, specifically in this case, that being into the muscle, is very important in terms of where we think we can be for efficacy as well as repeat dosing. And similarly to the exon skipping, where we saw persistent exon skipping at the end of the month, we also saw persistent concentrations of EDO51 in the muscle tissue, which supports our thinking that we should see clinically relevant levels of accumulation with repeat dosing. And that's really great for patients who will be on this therapy for long periods of time. And then in terms of the safety piece, we learned a lot from this study. What we saw was that EDO51 was generally well tolerated, and that all healthy volunteers in this study completed the study, which I think is a very important point. Any treatment emergent adverse events all resolved within the study time period. They were all mild and did not require any intervention on our part to treat the treatment emergent adverse events. And specifically at the 10 mg per kg dose, we see that all of those treatment emergent adverse events were mild. At the highest dose, at the 15 mg per kg dose, we did see some changes in kidney biomarkers. These changes in most healthy volunteers resolved within 24 to 48 hours post-dosing. There were 1 or 2 healthy volunteers that required a little bit more time to resolve, but those did resolve as well. I think importantly, though, to note there were no clinical sequelae that was noted during the time course, and no long-term effects on kidney function. And there were some transient changes as well at the 10 mg per kg dose, as those also resolved without any intervention within 24 to 48 hours. I know for this class of drug that people have been focused on the hypomagnesemia. We did see 2 individuals that had mild to moderate hypomagnesemia. Again, those also resolved without intervention. So based on this data set, we believe that we have the potential for a transformative therapy for PGN-EDO51 to be transformative for Duchenne muscular dystrophy, and that it is generally well tolerated with changes in kidney biomarkers that are measurable, monitorable, and reversible, as well as transient.
Excellent. Well, congratulations. That is quite a bit of really exciting and promising information. So
we're very excited about it, Erin. We're especially for the patients, and we're excited not only for those that have mutations that are amenable to exon 51 skipping, but also what it means across the platform. And I should also mention that the concentrations that we saw in muscle were also clinically relevant in terms of what we see in our preclinical studies related to our myotonic dystrophy type 1 program. And what we had seen in terms of the concentration is that we were able to achieve concentrations in the muscle that are clinically relevant for that program as well. So we believe that there's some translatability here across our platform.
Okay, excellent. I want to take a step back and talk broadly about oligonucleotide therapies, excuse me. And so I'm hoping we could do that for a little bit here. First, let's talk a little bit about how the delivery differs from other therapies in genetic medicine, specifically such as, say, gene therapy. Can you talk us through that a little bit?
Yeah, so I'd even like to take a further step back and talk about those exon skipping therapies that are currently available. And then we can weave in the gene therapy if that's okay.
Absolutely.
So oligonucleotides themselves actually modify gene expression. And so whereas gene therapy actually replaces the abnormal gene. And so with the current oligonucleotide therapies available for those exon skippers, what is being delivered is what we're calling naked oligonucleotides. So they don't necessarily have anything attached to them. You're just infusing the oligonucleotides. And as I said, you know, the inherent pharmacokinetics of oligonucleotides is not that great for delivery into those tissues of interest. We know that they are highly distributed to liver and kidney, but it's a bit more difficult to get them into muscle, smooth muscle, the heart. And this is a known challenge of the field. And what makes the technology here at PepGen different from what is from those other exon skippers is what we do is we take those oligonucleotides and we conjugate it to our proprietary peptide. And it is this proprietary peptide that confers the ability for better delivery. And our peptide essentially consists of 3 domains. It has 2 arginine-rich domains, and arginines are positively charged, and that positive charge is spread out by non-natural amino acids, and they flank a hydrophobic core in the middle. And it's this configuration of this cell-penetrating peptide that we believe confers better deliverability of the oligonucleotide to those tissues of interest. And what we have seen preclinically is that we do, in fact, have increased delivery or better delivery than other oligonucleotides, and to both striated muscle, to smooth muscle, to the heart, as well as get some penetration into the central nervous system. And this is and it's important for all of the diseases that we are focused on currently. In terms of gene therapy, the gene therapy again replaces the defective gene and in the DNA, whereas oligonucleotides are more acting with RNA to modulate that expression. And gene therapy is a wonderful advancement in medicine, but also has its challenges too, in the sense that there is a significant side effect profile associated with it. Usually, in order to deliver a gene in gene therapy, you have to deliver it within a virus, such as adenovirus, and these things can confer significant side effects, such as immunogenic effects, as well as cardiac effects, and additionally, and we've unfortunately witnessed some complications and mortality related to the gene therapies. So, that being said, the gene therapies are a wonderful advancement, and I can foresee, and others have also endorsed this, is that there is room for both gene therapy and oligonucleotide therapies, because gene therapy is sort of a one and done thing. So you can see that if you give gene therapy to a young boy with DMD, that as they grow, they actually turn over that muscle. And so those muscles that have been infected, if you will, with the gene therapy, either turn over or are cleared. And so as time goes on, the opportunity for redosing as they grow up is not a possibility. So you can see how gene therapy and therapy with oligonucleotides can be married, so that you can not only treat with gene therapy or pre-treat with oligonucleotides, but then later on have treatment with oligonucleotides to continue the benefit of the therapies themselves.
Yeah, that makes a lot of sense when you explain it that way. So now, and you mentioned cardiac tissue, and I want to talk a little bit more about that, specifically to reach, when we're talking about reaching specific type of tissue. So my question is, why is it difficult for cell and gene therapies to reach a specific tissue type, right? Especially tissues that are involved in these degenerative muscle diseases like cardiac tissue. So how is PepGen, and how is your enhanced delivery, oligonucleotides, how are they designed to overcome those challenges?
Yeah. There's been a lot of work done over the last couple of decades that the oligonucleotides, the naked oligonucleotides, have evolved to evade some of these mechanisms that break down the oligonucleotides. So oligonucleotides, like I said, when you infuse them, tend to go to the liver and the kidney. They're rapidly cleared. When they do get to a muscle cell, they can be broken down by endonucleases and other processes. And so over time, there has been modifications of the oligonucleotide to make them resistant to some of these clearing mechanisms, which are necessary, obviously, to protect our body from unwanted intruders. But in this case, we're talking about a therapy that is meant to confer a clinical benefit, and so there is a phosphorodiamidate morpholino oligomer that's been developed that is the most stable oligonucleotide that has been shown to be the most resistant to these types of breakdown mechanisms. However, it is still highly cleared by, kidney, and it still has difficulty getting into the tissue or staying in the tissue for whatever reasons. And sometimes these things are taken up into what are called endosomes and don't come out of the endosomes very well and then are degraded by the cell. We believe that by conjugating this very stable PMO to our cell-penetrating peptide, that we confer by this conjugation an additional stability to the molecule. So not only do we make it easier for the oligonucleotide to be taken up by the cells, but we also think that it is potentially that once it is in the cell, that it can potentially come out of the endosome more easily and go into the nucleus to perform its action of exon skipping and modifying that gene expression of dystrophin. In terms of the difficulty of getting into the different tissues, it could be that different tissues just have different mechanisms for degrading these oligonucleotides. I know that heart can be specifically challenging, and the thinking there is that perhaps maybe there is a specific challenge related to the heart because it's a single nucleated cell rather than striated muscle or skeletal muscle, which is a multinucleated cell. And so these are things that that the community is working on to understand so that we can confer better delivery. However, here at PepGen, we are also working to further understand why we have been so successful with the delivery of PGN-EDO51 to striated muscle and how we've achieved these high concentrations. Because unlike others, we are not dependent upon a specific receptor like a transferrin receptor. This is with the peptide. We believe that given the properties of the peptide, it just makes it easier for it to cross that cell barrier, or the cell membrane, I should say.
Gotcha. Okay. A pretty straightforward question I think I have here. Why are you and your team at PepGen, why are you specifically focused on rare neuromuscular diseases?
Mm-hmm. So we're focused on developing therapies that are transformative for rare diseases that have a high unmet medical need. Mm-hmm. Currently, the focus is on neuromuscular diseases, but we believe that this platform can be applied to many other diseases or many other rare diseases that can be amenable to this type of therapy. And so I think that at this point, we are focused on neuromuscular diseases. We believe that PGN-EDO51 will be transformative for DMD, and the learnings that we have from the neuromuscular program can be extrapolated potentially to certainly other neuromuscular diseases. As I said, we have our fast following EDO-DM1 program in myotonic dystrophy, followed by our other discovery programs for those other exon skippers in Duchenne muscular dystrophy. And we also have other programs in discovery that we are evaluating to apply the technology.
Okay, good. And I hope too that you'll come back and talk to us about those as well at some point soon too.
Absolutely.
Just one other question too. Talk us through the next steps for testing PepGen's enhanced delivery oligonucleotide therapies in patients.
Mm-hmm. So we are looking to start our clinical studies, our phase 2 clinical studies in DMD boys and young men in the first half of next year. And we are also looking to start our clinical studies, our phase 1 study in patients living with myotonic dystrophy as well in the first half of next year. And these studies will be developed to provide us with the data to support continued development of both therapies for these neuromuscular diseases, as well as to provide additional information to support continued development of the platform and to expand the platform to other disease states.
Okay, good. In your opinion, what techniques or methods perhaps might become more important in the analysis of oligonucleotides?
Right. So I think in neuromuscular disease, you're not always lucky enough to have a biomarker. For Duchenne muscular dystrophy, we have a pretty well accepted biomarker and looking at dystrophin production. For other neuromuscular diseases, we're not necessarily, it's not necessarily as clear. In DM1, we are looking at the correction of the spliceopathy, but that has yet to be discussed with regulators. And I think that those discussions are being planned with others. So I think it's really important, and not only to show the change, a clinically meaningful change on your biomarker, but also to have a good understanding of the functional benefit for patients. And certainly there are many clinical assessments that are employed in the neuromuscular clinical trials in order to understand or triangulate or measure the clinical benefit of a particular therapy. And I think that this is what regulators really care about. They really care about how a patient functions, feels, and survives, right? And so it's really important for us to understand what we are evaluating with our functional measurements. So things like the PUL or the North Star ambulatory assessment, and how that relates to a clinical meaningful change and how they feel, function, or survive. And I also think that it's important to really understand and go and ask patients. PepGen really puts patients at the center of everything that we do. We have discussed our protocols and our platform with patients, even at this early stage, to get their input to be sure that the clinical trials that we are designing are actually measuring things that are meaningful to them and that will make a difference in their lives. And so by understanding what is meaningful to them and makes a difference in their lives, it allows us to design those clinical trials to answer these critical questions as to whether a therapy is effective for that disease state, whether it be Duchenne muscular dystrophy, myotonic dystrophy, or something else. And so having their input is critical. And I think that as we are designing these functional studies, we really need to understand what is that minimally important clinical difference. What is that measurement, that threshold that we cross that really makes a difference in that patient's life every day? And so that may be being able to walk, X amount of meters, or being able to reach above a certain level. And I think that we need to understand that from patients. And then understand if we can measure it, and then have a conversation about its clinical meaningfulness. And I think that that is the most important thing, is understanding the function and the impact on function.
Perfect. Oh, thank you so much for all of this really great information. It's a topic that we haven't covered quite as much on Cell and Gene, and so this is really good information for our listeners and our readers. So thank you. We've kind of reached the formal end of our episode here, and so I like to ask my guests a question to get to know who they are when they're not in the lab or in their office. And so my question for you is, as I understand it, you live also in the Boston area?
Yeah.
Okay. So my question for you is, what is on your must-see to-do list for the greater Boston area? I always say in my next life, I'm going to, right out of school, go to Boston. So I've been there many times, and it's just a wonderful city. And I'd love to hear from your perspective what you think is like a must-see, must-do type of thing.
Yeah, I have to say the Freedom Trail is fantastic. I actually love going on that tour, and love seeing people dressed up and hearing about old-timey Boston, hearing about the molasses flood, seeing where it came up to on the buildings. So I really do enjoy that, and just walking around the city and understanding what the different buildings served in different times is a lot of fun. And I also like the graveyard part too. Seeing where different famous people are buried.
It is.
So it's a lot of fun to do on a fall day. Yeah.
Absolutely. It's a much like Philadelphia where I am. It's a city steeped in history. So it's just a wonderful just a wonderful part of the country. And I also saw that you are a graduate of a fellow Patriot League school.
Yes.
It's
Colgate.
Colgate. Thank you.
Yeah, I went to Colgate University. Yeah.
Yes. Yes. Well, that's wonderful too. So we have something in common. Well, listeners, that wraps up this episode of Cell and Gene, the podcast featuring PepGen's Dr. Michelle Mellion. Michelle, thank you so much for your time and for the actionable information you shared today. This was wonderful.
Thank you so much for having me, Erin. I really appreciate it.
Absolutely. So listeners, please visit Cell and Gene to register for our newsletter and to find more timely content to help you perform your role better. We'll talk to you soon.